So, ladies and gentlemen, you are welcome to my YouTube channel. Today, we are going to look at the concept of enzymes kinetics and reaction rates. And this PowerPoint is adopted from a previous version by Dr. Naimia. So, we are going to also make use of this PowerPoint and make this video lecture. So, in this video, this is what we are going to look at. We are going to look at the enzymes kinetics and reaction rate, the measurement of the rate of enzymatic reaction, the Michaelis Menten model concept, and the meaning of the constant Vmax, Km, and Kcat, linearization of Michaelis Menten constant, that is Lineweaver-Burk, phenomenon of cooperativity, kinetics deviating from the Michaelis Menten models, that is enzymes whose cooperativity models and allosteric enzymes, then protein ligand interaction. These are what we are going to look at in this video. So when we said enzymes kinetics, what is the enzymes kinetics mean? And why do you think it is important to measure or understand enzymes kinetics? So in this video, we are going to look at all that. So enzymes kinetics is the study of the rate of biochemical reaction by an enzymes. So when you say it's the study of rate of biochemicals, it's likewise enzymes, it means that how fast or how slow an enzyme is catalyzing substrates product, and that is enzymes kinetic. Let's say we have A plus B, the presence of enzymes to produce C plus D as the product. So when we said enzymes kinetics, it means how fast or how slow the enzymes can be actually converting the substrate here to product. And that is what we mean by enzymes kinetics. And that is what we're also going to look at in this video. So how important to understand enzymes kinetics is actually it usually provide us with an understanding how enzymatic efficiency is, and also it helps us to determine the concentration of the enzymes in a solution or in a biological sample. Assuming now you are given a blood and you are actually asked to calculate the kinetics of the enzymes, so that will give you an idea how efficient the enzymes is in a biological sample or in the blood, and what are the concentration of the enzymes in that given samples. We do that in biochemistry. And then when you set the rates that the enzymes kinetics trying to measure is determined by the change in the concentration of the substrate or product per unit time. Remember that in our previous class, we said that when you have an enzymes converting into product, the substrate usually, let's say this is your container, and in this container you have balls like this, and let's consider them as our substrate. So if the reaction starts, these balls, they will start deciphering, will have something like this. The blue balls will actually start deciphering to start generating the red balls. So it means that, let's say the reactant is the blue ball and the product is the red ball. So as a time goes on, the blue ball will be deciphering while the red ball will be appearing. So that is how it will continue moving. As a time goes on, the red color balls will still be appearing, and as a time goes on also, the blue color balls are also disappearing. So it means that as a reaction going on, the concentration of the reactant will be decreasing while the concentration of the product will be increasing. And when the reaction reach completion, then all the blue balls will disappear as a reactant, while all the red color will be appear. So that means that the reaction reach completion. So if you now want to measure the rate of the reaction, that is why to measure the rate of the reaction, we use a formula. To measure the rate of the reaction, we use a formula. It can be decrease in the concentration. It can be decrease in concentration of the substrates, or increase in the concentration of the product. So it can be decrease in concentration, it can be decrease in concentration of the reactant, or increase in the concentration of the product per unit time. And that is how we measure rates of the reaction. And I hope it is clear. Let me use it. So to measure rate of the reaction, it is equals to the decrease is going to be decrease in concentration. It's going to be decrease in concentration of reactant per unit time divided by time. So this is actually one way of measuring the rate of the concentration or the rate of the reaction, either decrease in concentration of the reactant per unit time, or increase, or it can be increase in concentration of product per unit time. So this is the two ways of measuring rates of the reaction. So then, of course, the determination of rate of biochemical reaction catalyzed by an enzyme can provide an information about the presence of activators or inhibitors, or if there is a tissue damage. Like for example, maybe you know that actually from the literature, maybe the turnover number of the enzymes, maybe it's catalyzing maybe 20, catalyze, let's say 20, let's say it catalyzes 20 substrates product. Let's say it catalyzes 20 substrates. It catalyzes 20 substrates to 20 product. So now, you know this from the literature. And now maybe after you know that, okay, you have the enzymes in a given biological samples, and you now realize that, okay, so now the enzymes is now, instead of converting 20 substrate to 20 product, it's now converting 30 substrate to 30 products. So what does that mean? It means that there is an activators. And what is activators? When you said enzymes activators, it means that there are molecules that increase the activity of the enzymes. And we also have enzymes inhibitors. So when you said enzymes inhibitor, what does that mean? And enzymes inhibitors are the molecules that decrease or inhibit the activity of the enzymes. Let's say here it is converting 20 substrates to 20 product, and now maybe in the presence of inhibitors, then it's now producing, or is now converting 5 substrates to 5 product. So what does that mean? It means that definitely in that given samples there is an inhibitors, as there is a molecules that slow down the activity of the enzymes. And you can also check if there is a tissue damage. So a tissue damage, actually, assuming now you have an enzymes, the enzymes is in the liver, you have an enzymes in the liver, and maybe in the blood, it's known that, it is well known that maybe the concentration of your enzymes, let's say, is 1 milligram per mole of the enzymes. And now, after some time, then you now check maybe in the blood, then you now see 5 milligram per mole of the enzymes. So what are you going to do, or what will that suggest? It means that it's supposed to be in the blood is 1 milligram, and now you check that is 5 milligram per mole. So what does that mean? It means that the organs that contain that enzyme is damaged, and that is why there is a release of that particular enzymes. And that is why sometime in medicine or in the normal medical laboratory test, where there is what you call an enzymes function test. If there is any problem with the liver, there are some specific enzymes that they are supposed to be in the liver, but if you check them and you find out that they're in the blood, it tells you that the liver is having problems. There are some proteins or enzymes that are supposed to be in the brain, and now you get them in the blood. So that is to tell you that the brain is having problem, it damaged, and that is why it released its contents. So the rate of the reaction can help to determine whether there is an inhibitors or activators, or if there is any tissue damage. So this is actually very important knowledge that enzymes is always trying to pass. Okay. Yeah, rate of the reaction, that is the enzymes kinetics. Then this study of how an enzymes respond to experimental parameters can be used to design inhibitors, design activators, or modulate activity. So with this knowledge of enzymes kinetics, if you are able to get this knowledge from here, you would be able to design an inhibitor or a compound that can serve as a drug. Like for example, let me just give this example in terms or in the case of rational drug design discovery. In the case of rational drug design discovery, assuming now you identify a particular enzyme that is important for or that is associated with the disease development. So you can design inhibitors or molecules that can either decrease the activity of the enzymes or increase the activity of the enzymes. So let me give this example. There are what we call arachidonic acids. There is arachidonic acid in the body, and the major functions of this arachidonic acids is actually, let me write it here, arachidonic acid is an amino, is a fatty acid, and of course this arachidonic acid is involved in the synthesis of prostaglandins. And what are these prostaglandins? These prostaglandins are the molecules that are associated with pain and fever, and some other inflammatory reactions in the body. So these prostaglandins, if we have pain in our body or fever, is due to production of these prostaglandins. And these prostaglandins are produced by cyclooxygenase 1, that is COX-1, cyclooxygenase 1, and cyclooxygenase 2. So these are the two enzymes that produce these prostaglandins. So with the knowledge of kinetics of enzymes or rate of the reaction, we will be able to design a molecules that will inhibit the activity of the enzymes. And once you do that, then it will block the formation of the prostaglandins. And of course, you will actually cure the fever and the pain. Fever and the pain will be relief. And that is what happened. Aspirin is a drug, we know. Aspirin is a drug that actually, so this drug is an inhibitor of COX-1 and COX-2. That is why if you use aspirin, you can easily relieve pain and fever because it blocks the activity of these two enzymes. Therefore, it would either stop the formation of prostaglandin or slow the formation of the prostaglandins. And that is how important and how essential understanding the knowledge of enzymes kinetic is. So then in the next this thing is, so enzymes kinetics at the same time is also the enzymes kinetics and reaction rate is the best way to study the mechanisms of action of enzymes, and it's also to determine the reaction rate and also the change in response to experimental parameter, and this is studied in enzymatic kinetics. So ladies and gentlemen, you should understand that this knowledge of enzymes kinetics is also it helps in determining the mechanisms of action of a particular enzymes. So the best way to determine the mechanisms of enzymes action is to understand the kinetics and the rate of the reaction. So like, for example, let's look at this. This is like an enzyme. So in this case, it's like you are increasing the substrate, and then of course the rate of the reaction will be increasing. So this is just to show you that this is a graph that's trying to show how the enzymes is involved in converting substrate to product, and as you increase the concentration of the substrate, also the rate of the reaction will increase. So what is enzymes kinetics? Enzymes kinetics is determining the rate of enzymes control reaction and study how it changes. That is because the word kinetics, enzymes molecules that catalyze the reaction. And then kinetics is how fast or how slow the reaction is. So enzymes kinetics is to determine the rate of an enzyme's control reaction and study how it changes. So the changes also here can serve as the kinetics. So enzymes kinetics is the study of how enzymes catalyze the rate of the reaction and how it also changes, maybe by increasing the activity or decreasing activity due to addition of activators or inhibitors. So that is enzymes kinetics. And then, of course, so this is actually how the reaction usually takes place. So here we have an enzymes, and of course, we can see clearly here, this is the substrate, and this is the enzymes. And of course, this is the active site of the enzymes, as you can see from here. As you can see from here, so this is actually how we have the enzymes plus substrate. So the substrate will now bind to the active site. The substrate will now bind to the active site, form an enzyme substrate complex. And then, of course, the active sites of the enzymes will now start catalyzing the substrates into products. It will now start catalyzing the enzymes. It will now start catalyzing the substrates into product, and then we will now have the product released. So this is what happened. So usually during the course of the reaction, enzymes is converting substrate into product, and then the enzymes will now recycle. So usually the enzyme do not consume the cause of the reaction. So we have an enzymes plus substrate, and then the substrate will bind to the active site to form what you call enzymes substrate complex. And then, of course, the active site of the enzymes, or the enzymes will now start converting the substrate into product to produce enzymes product complex. And then finally, the products will leave the active site of the enzymes, where the enzymes will also go back and catalyze another substrate into product. And also in addition, so how is enzymes activity measured? How do we measure the activity of the enzymes? So we have it, this V stand for the rate of the reaction, and we have minus D into concentration of S. So the minus here means decreasing. It entails the decreasing, because this S stand for substrate. So as the reaction is progressing, then the concentration of the substrate will be decreasing with time. So that is why here we have minus D into concentration of S divided by the change in time, that is dt. Then it's equal to, so in the case of product, because the product is, we are forming the product, so it should be positive, while the reactant, which is the substrate, is decreasing. So that is why it is negative. And in the case of product, because it's the product you are forming, and that is why you have delta P all over delta T. That is change in time. That is change in product divided by the change in time. So that is actually how enzymes work briefly. So now this is actually what happened. We see here we have an enzyme. This is, in this case, we have our substrate here. We have our substrate here, and the substrate now, and this is the active site, and then the substrate bind to the active site. So we have the enzymes bind substrates here, and then of course the enzymes release product after the product is actually fully formed. So this is how the kinetics is. We have an enzymes plus substrate to produce enzyme substrate complex. So this is usually produced. And then finally, we'll have the free enzymes and the product. So the product will be released. And then of course, so this is how measure the kinetics of the enzymes, or the activity of the enzymes, by decreasing the concentration of substrate over change in time, or it can be, or you can use this to measure, only this to measure the activity of the enzymes, or you can decide to say, okay, you can measure the rate of the formation of product with change in time. So by measuring the disappearance of a substrate or emergence of product, so in the presence of very small quantity of enzymes, let's say the quantity of the enzymes is 10 raised to the power of minus 8 molar, 10 raised to the power of minus 8 molar. So if you have this, so in the presence of a small quantities of enzymes, let's say 10 raised to the power of minus 10 and 10 raised to the power of minus 12 molar. So quantities that are known to be catalytic. So this is a quantity that are known to be catalytic. So under optimum condition of pH, temperature, and the presence of cofactors etc. So by saturating conditions of a substrate are used, then you can also do that by determining the initial reaction rate, that is the initial velocity. So this is tell you how is enzymes catalyzed reaction is measured. So there are different ways of measuring the enzymes catalyzed reaction. Number one, you can measure the enzymes catalyzed reaction by the difference of, you can do that by measuring the difference of substrate or the emergence of product. That is the one way to do that. And then the second way, the presence of small quantity of enzymes, let's say 10 raised to the power of minus 8 and 10 raised to the power quantities are known to be catalytic. So you can actually have an idea of the quantities of the enzymes that are known to be catalytic. So if you know that, then you can use that to measure the activity of the enzymes. You can also measure the enzymes activity by under the conditions of optimum pH, temperature, and presence of cofactors, because we know that enzymes generally work at a particular pH and temperature, and also cofactors. So if the pH is altered, if the temperature is altered, or in the absence of the cofactors, the enzymes might not work. So under optimum conditions, we usually use the optimum conditions or pH, temperature, and of course the presence of the cofactors to measure the enzyme's activity. And you can also do that by measuring or by saturating the conditions of substrates that are used. Like for example, you can, if you know that, okay, this is the enzyme, so you can supply the enzymes with enough substrate. You can supply the enzymes with enough substrate so that there will be a maximum enzymes activity. So you can do that when you are trying to measure the activity of the enzymes. Or you can do that by determining the initial rates, the initial reaction rate, which is actually V naught. So you can do that. So these are the ways that you can use to measure the enzyme's activity. So all this must be maintained. All this must be actually put in place before you measure the activity of an enzymes. So in addition here, we are going to look at, of course, how the enzymes and the substrates work. So you see we have an enzymes here, and then the enzyme binds with the substrate. Then there is, of course, the enzymes release the product. So what happened? So generally, when you are trying to actually in the kinetics of the enzymes, you see this is the reaction rates and this is the concentration of the substrate. So this is the concentration of the substrate. So generally, if you have an enzymes, as you increase the concentration of the substrate, as you increase the concentration of the substrate, of course, there is an active side. So increasing the concentration of the substrate also leads to increase in the rate of the reaction. So therefore, we should know this, and it is very, very important in measuring the rate of enzymatic reaction. There is an active site, and also the active site is for the substrate to bind. So as you have the enzymes concentration, you are adding substrate. So as you are increasing the concentration of the substrate, then the rate of the reaction will increase. And that is why if you look at here, that is what we have. This is the concentration of the substrate. This is the concentration of the substrate. And this is the reaction rate. This is the reaction rate. So if you are increasing the concentration of the substrate in this direction, then the rate of the reaction will also be increasing. As you are increasing the concentration, of course, the rate of the reaction will be increasing. until when it comes to a stage where there is no available active site for the substrate to bind. So if you have enough concentration of the substrate and there is no enough active site for the substrate to bind, and that is how we reach this stage. It will come to a stage where there will be no increase in the rate of the reaction. And that stage is called the Vmax. That is, it means that the enzymes reach its maximum velocity. It means that the enzymes reach its maximum velocity. If the substrate doesn't get an active site of the enzymes to bind, because the active site is already saturated with the substrate. So that is how we have this graph. We call it hyperbolic graph of Michaelis Menten. So as you have the enzymes, look at it like in this case, you have enzymes bind to the substrate in this case. So if there is no more, and you have, let's say you have so many other substrate here, you have so many other substrate here, but there is no active site. So the rate or the speed of the reaction will remain constant. But in the case where, okay, you have the active site and you have some of the substrate bind to the active site, and you are still including the substrate, and of course, the other enzymes will continue binding to the active site of the enzymes. And once that happens, it means that there will be no increase in the rate of the reaction, because all the active site is completely saturated with the substrate. And that is how we have this. So it means that the enzymes reach its maximum velocity. That is the meaning of Vmax. That is maximum velocity. And we have a stage where Km, this is Km, this is Km. So you see from here, it means that if you look at it, as the substrate concentration increase, of course, at this stage, the activity of the enzymes also, or the rate of the reaction is also increasing. So at this stage, we have Km equals to half Vmax. Km, it means that this Km, as we have it here, Km is equal to half Vmax. What does that mean? It means that the active site of the enzymes is half filled with the substrate. Is half filled with the substrate. And if we have Vmax here, it means that the active site of the enzymes is completely filled with the substrates. And that is the meaning of Vmax. And of course, Km is equal to half Vmax. It means that half Vmax, it means that the active site is half saturated with the substrates. So that is this. And of course, so the measurement of the rate of the reaction still can be, so as the reaction progress, you see at this point we have the concentration here and we have time. So as you increase the concentration, so the rate of the product formation is directly proportional to the surface of the substrate. So it means that as you are increasing, as the reaction progress, the concentration of the substrate will be decreasing, while the concentration of the product will be actually increasing. That is why we should understand that, of course, during the course of any chemical reaction, whether in chemistry or biology, as the concentration of the product increases, of course, the concentration of the substrate will be decreasing. So if the substrate is completely exhausted, it means that there will be zero concentration of the substrate and there will be 100% concentration of the product. So the rate of product formation is directly proportional to the surface of the substrate. So you can say that, of course, appearance, we can now take it as this, that appearance of product, appearance of product concentration is also directly proportional to this appearance of the substrates. So that is what we have. So to define the rates of the reaction, we need to define the steady state where we have enzyme substrates is constant. So that is why here, when you have an enzyme plus substrate, then it is going to give us enzyme substrate complex. So when you have an enzyme substrate complex, it is reversible because the substrate, the enzyme substrate complex can still dissociate to give back free enzymes and free substrate. So in the forward reaction, the kinetic constant is K1, and in the reversible process, the kinetic constant is minus K1. And of course, the enzyme substrate can also break down to produce free enzymes plus free product. And there we have K2. So to define the rate of the reaction, we need to know the steady state where enzyme substrate is constant. So if you can look at this graph, this is the reaction progress, where we have the concentration here of the product, and of course, here we have the substrate, and of course, this is a time. So as the reaction progress, there is a formation of a product, and of course, there will be a decrease in concentration of the substrate. So that is why at this stage, V is 0, and of course, at this stage, V is 0. So if the action reach completion, it means that the product is completely formed, then of course the velocity will be 0. And in the case of substrate, when the substrate is completely exhausted, then the velocity will also be 0. So that is why the rate of the product formation is directly proportional to the substrate disappearance. So the next thing is Michaelis Menten model. So in Michaelis Menten model, this is the Michaelis and this is the Menten, and they are the one that actually developed Michaelis Menten equation. So in a Michaelis Menten equation, it governs by enzymatic reaction, and most enzyme exhibits Michaelis Menten type kinetics, in which the graph of initial velocity V naught versus substrate concentration. So the B naught is stand for the rate of the reaction, so the initial rate of the reaction. So as you increase the concentration of the substrates, this is of course increasing the concentration of the substrate, and this is the V naught. So as the concentration of the substrate increase, of course the rate of the reaction will be increasing. So at this stage, you have Km be equals to half V max. And of course, as the substrate concentration increases, it will be binding to the active site of the enzymes. So it will reach to a stage where the active site is fully saturated with the substrate. So at this point, the reaction will now remain constant. It will remain constant, meaning that there will be no increase in velocity. So when we have this, therefore, now if we look at this reaction, we have enzymes, substrates, we have enzymes plus substrate give us enzyme substrate complex, and the kinetic constant for the forward reaction is K1, and the kinetic constant for the reverse reaction is minus K. So, and of course, for the kinetic constant of enzyme substrate complex to enzymes plus free enzymes is K2. So therefore, the rate of this reaction is now going to be, of course, the rate constant for the final reaction is going to be K2 into the enzyme substrate concentration. We know that if we are going to get the rate of the reaction, it is always we measure the concentration of the reactant. So, of course, according to the law of chemical kinetics, the rate of the reaction is directly proportional to the concentration of the reactant raised to the power of n. The n stands for the order of the reaction. So, but in this case, if you want to get the rate of the reaction here, so the rate of the reaction, which is V here, is directly proportional to the concentration of enzyme substrate complex, and K2 here stands for the rate constant. So to get the Km of this reaction is now going to give us Km is Michaelis Menten constant. We are now going to take K1 is K1, sorry, minus K1 plus K2 all over K1. It will give us the Michaelis Menten constant. So after we have the Michaelis Menten constant, then the next thing now is, because this is the reaction, so we're now going to have enzyme K1 to measure the rate of the reaction here, measure the rate of the reaction. So it's going to be, of course, K1 is going to be K1 okay, it's going to be K1. Let me have it like this. Okay. It's going to be something like this. So to measure the rate of the reaction, which is K, sorry, which is V here, it's now going to be K1 into enzymes concentration times substrate concentration. And this is actually for the forward reaction, And for the reverse reaction, it's going to give us, of course, for the reverse, this is for the forward reaction. VF here is for the forward reaction. And then for the backward reaction, which is VB, for the backward reaction, the rate of the reaction is going to be minus K1 into enzymes substrate complex. So that is why this is the rate of the reaction for the backward for the enzyme substrate complex formation, and this is the reverse reaction for the formation of enzyme substrate, and of course plus K2, that is for the formation of product. And of course, after we have this, then we can proceed with this. So we are now going to make the ST subject of the formula. So here we have enzyme substrate complex concentration, and here we also have enzyme substrate complex. So we are going to now make enzyme substrate complex subject of we are now going to factorize it. So after you factorize it, like here we can have enzymes substrate complex like this. Then we are now going to open bracket, we have minus K1 plus K2. So we have this, and of course here we have enzyme substrate. So what we are going to do next is to actually make enzyme substrates complex subject of the formula. And that is how we arrive at enzyme substrate complex like this. It will be equals to K1 all over minus K1 plus K2 times enzyme substrate complex formula, enzyme substrate complex. And of course, from there, you can now make, and of course, we know that the Km is minus K1 plus K2 all over A1 from our previous slide. So this is our Km, that is Michaelis Menten constant. So since we have this, we can just substitute the whole of this with Km. So if you substitute the whole of this with Km, we are now going to end up of having enzyme substrate complex times substrates divided by Km. So that is how we arrive at this. And then from there, we have the initial rates to be this. And of course, finally, we are going to arrive at this after dividing everything. So we are going to have V naught is equal to V max all over substrate concentration all over Km plus substrate concentration. And this is what you call Michaelis Menten equation. So this is how Michaelis Menten equation is actually is being derived. Yeah. So from here we have Michaelis Menten constant. So what is the value? What is the meaning of V naught, V max, and Km? So we are going to look at it in the next slides. So after this, we now have already as our Michaelis Menten equation. And of course, Michaelis Menten equation is usually represented in in a graph, in hyperbolic graph. And this is where we have our B naught, that is initial rates. And of course, as the concentration of the substrate increases, then the rate of the reaction, which is the initial velocity, will also be increasing. So that is what we have here. So as the rate of the concentration increases, then the Km, the substrate concentration increases, then the rate of the velocity will also be increasing, or the rate of the reaction. And of course, if the enzyme is completely filled with the substrate at the active site, so no more substrate will be accommodated. So therefore, the rate of the reaction will remain constant. And that is why we have this hyperbolic shift of the Michaelis Menten equation. So this is the Vmax substrate concentration all over Km plus substrate concentration. So let's look at what is the meaning of Vmax. So the Vmax here, it is the theoretical maximum velocity that is never reached and is constant. So it is a maximum velocity that is hardly for an enzyme to reach. And it requires a very high substrate concentration and for all enzymes molecules to be bound to the substrate. So when you have Vmax, it means that all the enzymes is bound, all the active site of the enzymes is already bounded to the substrate, and that is how the enzymes will reach its maximum velocity or maximum speed. So in the Michaelis Menten equation representation, the curve of V naught, which is the rates versus substrate concentration, is asymptotically loss. So we have it here. This is where we have the maximum velocity, and this is where we have, let's say we have two different enzymes. So we have enzymes 1 and we have enzymes 2. So you see this is the Km for enzymes 1 and this is the Km for enzymes 2. And this is the substrate concentration. And of course, this is where the Vmax for the enzymes 1 reach, and this is where the Km the Km for the enzymes 2, where we have half Vmax. So sometimes you can actually plot the Michaelis Menten equation or the Michaelis Menten graph, compare the activity of the enzymes, because Km is one of the kinetic parameters that is used to tells the kinetics behavior of an enzymes or the catalytic activity of an enzymes. We'll also look at it. So the meaning of Km. So the substrate concentration at which the rate of Vmax is over 2 is reached, that is Km, and the Km might range in values from 10 power minus 1 to 10 power minus 6 molar, depending on the enzymes. And the value of the Km for an enzymes depend on a particular substrate and on other variables such as temperature and pH. So you see, the value of Km depend on substrate concentration, temperature, pH, and so many other factors. And then the Km establish the approximate value for the intracellular level of a substrate. And of course, the reaction rate is very sensitive changes in substrate in the Km range. And of course, the Km is usually related to an enzyme's affinity for the substrate. So the Km value is also show how an enzyme is having affinity to bind to a substrate. And the lower the Km value, the higher the binding affinity of the enzymes toward the substrate. So that is why the lower the Km value, the best the enzymes is, and the higher the value of enzymes, the higher the value of Km, it means that the less the affinity of the enzymes toward the substrate. So it is always good to have an enzymes with lower Km value. It is always good to have an enzymes with lower Km value. And ladies and gentlemen, if you didn't join my YouTube channel, please try to join my YouTube channel as a membership. So in the next video, sorry, of course, so the meaning of Km still on that. So example, let's say you have hexokinase and glucokinase. They have different affinities for glucose. So Km for hexokinase is very low at 0.1 molar. And this works for glycolytic purpose when glucose concentration is low. So if the glucose concentration is very low, this is where the Km value will play a role in catalyzing the glucose for glycolysis. But of course, for the Km value, for the Km value of glucokinase is 5 millimolar, and the major functions of this Km value, the functions at high concentration for glucogenic storage purposes. So it is actually important for gluconeogenesis. So when the concentration of the glucose is higher than the required value in the blood, then of course the glucokinase will now pick off and use the glucose for gluconeogenesis because the concentration is very high. So that is when the glucokinase will recognize the glucose and start converting it into gluconeogenesis. Sorry, into glycogenesis, the synthesis of glycogen. And of course, it is very, very important. So the Km value and the Km value of hexokinase and glucokinase are different. And the lower the Km value, the higher the affinity. So here it means that hexokinase have higher affinity to glucose than the glucokinase, which have the Km value of 5 millimolar. And in addition, they have different purposes. They have different purposes. Yeah. So then the next thing is, of course, okay, this is the Michaelis Menten constant where we also talk about the hyperbolic shift of glucose. So the measurement of the rate of enzymatic reaction, which is the Michaelis Menten. So the explanation of experimental data with the Michaelis Menten equation. So it's substrate, when the substrate concentration is very low, then of course the Km, which is V naught. So let's see here. So when the substrate concentration is very low, then S concentration is negligible versus the Km, and then V naught increases linearly with the substrate concentration. So if this substrate is neglected here, then it means that the V naught is now going to be V max all over Km times substrate concentration. Yeah. So at this point, so but when we have substrate concentration to be very high, then V naught is equal to V max if the concentration is very high. So you need to understand this scenario. If the substrate concentration is very low, then the substrate concentration here at the denominator will be neglected. And at the end, we are going to have V max times substrate concentration all over Km. That is what we are going to have. And of course, when S is very high, then the V naught is going to be V max. And if V naught is equal to V max over 2, then at the end, Km is equal to substrate concentration. So you should understand this scenario. So what is enzymes turnover number? So an enzymes turnover number, it means that it's a number of substrate molecules that are converted to product by unit time. So the per unit enzymes per unit time under substrate saturation conditions. So it's the amount of the substrates molecules that are converted to product per unit enzymes molecule per unit time under substrate saturation condition, and K cut represent turnover number. And then this turnover number is the same thing as maximum velocity all over enzymes concentration. And it has a unit of per second. So now the next thing is Lineweaver-Burk. So Lineweaver-Burk is a reciprocal of Michaelis Menten. Of course, so if we take the reciprocal of Michaelis Menten, then it is going, when you said reciprocal, it means that you are going to take the numerator to be the denominator and the denominator to be the numerator. Let me just actually show how it is going, how it's going to work. Remember that in the formula is V naught into V max times substrate concentration all over Km plus substrate concentration. So if we take the reciprocal of this, it means that it's going to be 1 over V naught is equal to Km plus substrate concentration all over V max and substrate concentration. So when you rearrange this equation, when you rearrange everything here, it is going to give us 1 all over V naught is equal to Km all over V max times substrate concentration plus, of course, here 1, okay, S, that is substrate concentration all over V max times substrate concentration. So this substrate concentration will now cancel this. So that is how it's left with 1 all over V max. And that is how we have this. So we transform the what you call Michaelis Menten to Lineweaver-Burk plot. So Lineweaver-Burk plot is like, for example, in the case of Michaelis Menten, the graph is hyperbolic. But in the case of Lineweaver-Burk, when you transform the equation by taking its reciprocal of the Michaelis Menten, then the graph is going to be linear. So why do we need linear graph? So we are going to look at the reason why. So it is the inverse of representation of V naught versus substrate concentration in the case of Michaelis Menten equation where we have hyperbolic. So, but in the case of Lineweaver-Burk, it is actually instead of V naught against substrate concentration, so it's now going to be 1 over V naught against 1 over substrate concentration. And of course, this is where we have the intercept here is 1 over V max. This is the intercept. And then of course, this is what we have in our x-axis. So now the intercept, so this is useful for calculating Km and Vmax more accurately. So it is used, the Lineweaver-Burk, it is used to actually calculate Km and Vmax more accurately and the mechanisms of the action of the enzymes inhibitors. So it is the best way to use this. And the intercept on the x-axis is equal to 1 minus Km. Yeah, the intercept on the y-axis is minus 1 over Km. Then of course the y-axis intercept is 1 over Vmax. At the y-axis is 1 over Vmax. So the slope, the graph is going to be m, that is Km over Vmax, that is the slope. So what is the purpose of the inverse? When V naught is plotted against substrate concentration, it is not always possible to determine the Vmax. So it's not it's not always possible to determine when Vmax has been achieved due to a gradual upward slope of the curve at this substrate concentration. So that is a major reason, because the time the Vmax is problematic. Okay. So then that is actually for the Lineweaver-Burk. And so the next thing is actually cooperativity, and which we are going to look at it in the next video. And if this is the first time you are going, please try to join to my YouTube channel. Thank you.